Hello, good afternoon, everyone. Hope you guys are all doing good. Um, so the marks for the quiz one is already up. The class FH is pretty good. It's actually comparable with other sessions as well. So keep working hard and studying from the study guides and review questions, and keep up the good work. And there's another quiz coming up in 2 weeks, I think. But we'll talk more about it as it approach. Couple of things that are coming our way. Let me just pull up our schedule and tell you about that. This week we will be finishing up um, lecture 4, and then next week there are no lectures. You just have to work on your Beetroot lab, which I'll explain on Thursday's class. Okay. It's a very straightforward lab. You do it online. There are some questions and some videos that you have to watch and complete, and you will hand it in on Thursday. It will be worth 10% off your grade. The next quiz will happen on October 8th, and it's going to be lecture 3 and 4. Okay, so I'll remind you again that there are no lecture next week. You just have to use the time to finish the lab. We do have a bit of time today, so I think we're just gonna slow down a little bit and maybe do a little bit of review for lecture 3 before diving into lecture 4. So there is a diagram that looks like this in the lecture 3 study guide. If you have the study guide with you, you can turn to this page and we'll label the cell together, and I'll remind you, of the functions of some of these things that we have in the cell. There we go. I'm just gonna make a copy of it, and I'm actually gonna put it in today's class notes, okay? You guys are 202, today is the 24th, so here we go. All right, so this is an animal cell. Animal cells are usually more spherical in shape as opposed to if you look at a plant cell, they are more rectangular in shape because of the cell wall. So let's label some of these things here. And I'm not doing it in any particular order, just writing them out as I see fit. So for example, over here, actually you guys can help me maybe. Can anybody tell me what this structure is over here on the left side?
Mitochondria.
That is the mitochondria, Micah. Thank you. So let's write that down. Okay, so over there. And mitochondria is the site of energy production. Okay, so specifically, it is where part of cellular respiration occurs. Okay, so let me just remind you the formula for cellular respiration. Even if you don't have the diagram with you, you can still write some of these things down to help you remember it, okay? So that's glucose plus carbon dioxide, which plus oxygen rather, which produces carbon dioxide and water. And more importantly, it is going to produce energy. Right? So that is the mitochondria. All right. Here's another one. And of course, the number of mitochondria is going to be different in different cell types depending on the energy requirement of that cell. Okay, so we have another structure here. This whole thing. Does anybody know what that might be?
The Golgi.
That's right. That's the Golgi body or Golgi apparatus. Sorry, guys, I just had to restart the program. It's has been acting up a little bit lately. I'm not sure why. Let's see if it works now. There we go. Okay, so that is the Golgi body. Now on the quiz, the second one, okay, there will be some labelings that you have to do. Okay, so I'll show you an organelle, and you will have to type the name of the organelle. Okay, so you can say Golgi body. Golgi body is fine. Okay, you don't have to put in bracket apparatus like I do here. I'm just showing you in case you read about it and you see a different name that you wouldn't be surprised. Okay, so Golgi body is part of the endomembrane system, and its job is to help sort, modify, and repackage vesicles. Okay. Proteins arriving in vesicles. Okay, so that's the Golgi body. Okay. How about this green little ball thing here?
Lysozyme.
Very good, Fahima. That's the lysosome. Okay. And lysosome is also part of the membrane system, part of the endomembrane system, part of the endomembrane system, and it contains a mixture of enzyme, digestive enzyme. So these enzymes can do various things, including digest large molecules, okay, lipids, carbohydrates. It can even digest an entire bacteria in some cases. And the lysosome can also trigger apoptosis, which you might remember from the notes. It's programmed cell death. Sometimes you need to deliberately kill off a cell as part of normal development, or if the cell is not behaving properly, or if it's getting too old, you might have to trigger the programmed cell death to destroy it for the benefit of the entire organism, right? Okay, so that's that. How about these stuff out here? Anyone know what that might be?
Yes. Ear.
Hmm Is it the smooth one or the rough one? What do you think? Let's zoom in a little bit to take a closer look.
It's the smooth one.
Yes, because they do not have the ribosomes, right? So that's the smooth ER. Okay, on the test, on the quiz, okay, please spell the whole thing out. Okay, smooth endoplasmic reticulum. Oh, by the way, some of you probably wondering like, can you see the quiz result, quiz one? We do not give back the quiz questions for online testings because we have to maintain the integrity of the uh, test bank. Right? We don't want the quiz questions to be like out there in circulation. So for that reason, we do not return the quiz questions. I know it's not ideal, but it's one of the limitations we have in an online learning environment. But sometimes if I see a questions where a lot of people get it wrong, then I'll share it with you guys and we'll take it up in class so that you can learn from it. Okay. All right. So smooth endoplasmic reticulum. The main function of that is to synthesize lipids Okay, they can also help break down lipids and detoxify drugs. Also part of the endomembrane system. Instead of writing it so many times, maybe I should just highlight all the stuff that is that belongs to the endomembrane system. Anyway, there it is. Okay, moving up a little bit. We have these things here, right? I'm sure if you recognize the smooth ER, you would recognize these ones as well. These are the rough endoplasmic reticulum. Okay. Also part of the endomembrane system. And what would the function be for the rough ER? Anybody? What does it do? What does it produce?
Protein.
That's right. So this is synthesis. Oh my goodness, is not working again. Not really sure why. Keeps on disappearing. Sorry. Let's try it one more time. Synthesis. Synthesize proteins. And package them into vesicles. There you go. Okay, more lysosome here. How about this one? We learned this briefly last week, I think, at the end. Anybody
So
remember?
centrioles.
That's right. These are the centrioles. I think it's pronounced centrioles, like the LE. Sometimes we want to say it, but I think it's silent. So this one is made up of, actually, we'll say it it's a pair of them. They produce spindle fibers. Spindle fibers, which help move chromosomes during cell division. Okay. And plants don't have these. They still have spindle fibers, but they are produced in a different way. But we do. We have the centrioles. Okay. There are some like random fibers here, right? You see some more here, some more here. Those are generally just cytoskeleton. Okay? You can't really tell which one they are, so I'm just going to put cytoskeleton here. Okay, we learned 3 types of cytoskeletons. We have the microfilament, okay, which is made up of actin. We have the intermediate filament. This one, it helps stabilize nuclear envelope, for example. And then we also have the microtubules, okay, which forms things like the cilia or the flagellum. And in fact, the spindle fibers here are also made up of the microtubules. Okay. This big circle in the middle, obviously, is going to be the nucleus. And what about the thing in the middle? of the nucleus?
Is it the nucleolus?
That is the nucleolus. There we go. Which makes ribosome for us, right? Synthesize ribosome. Okay, so of course the ribosomes can be stuck to the ER, but they can also be like floating around by themselves. I mean, you can't really see them maybe on my screen, but if you have the picture, you will see all these random dots here and there. Those are all individual ribosomes, okay? Free ribosomes, if you would. They too can produce proteins. Okay, so I hope that is a good review for you. Just try to help you remember some of the stuff that we've learned. Because my outdoor class is actually uh, not you guys are better because you guys have class on Tuesday and Thursday. They have class on Monday and Wednesday, and for the Monday class, they actually lose 3 days because of the holiday. There's like the Labor Day and Thanksgiving and things like that, and so we're trying to catch up with them, and you guys have more time, which is why we are able to do some review like this. So I hope that was helpful. Last time we did a drawing, which was the fluid mosaic model, right? We
Sorry,
talked about
just
how. Hmm?
a
Sorry, go ahead.
question.
Yeah,
So
yeah, please.
there's two more labels left, the cell membrane and the nuclear membrane.
Oh, yeah, yeah, yes. Thank you. So like cell membrane is right here, right? It's on the outside of the cell. I actually didn't look at all the words. But there you go. This is the cell membrane, which is what we drew last time with the phospholipids and everything. And the other is the nuclear envelope, right? Which is what surrounds the nucleus. This thing with all the pores on it, right? That's the nuclear envelope. It's hard to point to, so I'll just color it green here for you. And we'll say that is the nuclear envelope. Okay. Thank you for pointing that out. I know there is also a plant cell as well, but it's pretty much the same as this. Okay. But there is also the cell wall, the central vacuole, and the chloroplast that you can label on that one. Okay, so I was saying last time we drew the fluid mosaic model of the cell membrane. Looks something like this. And so we are gonna explore more about the cell membrane today. Okay, so let's see. There we go. I did that already. So first, this is a very important question. Okay, I'm gonna put a couple stars here for you. Okay, I'll read it to you first, and then we'll talk about it. How would the percent of saturated, unsaturated fat, and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in a hot environment? Okay. Maybe we can write this down, okay, in the class note for today. Let's see if the straight line function works. Oh, it works. Wrong color though. So let's do a table here. And then one more. We'll say this is cold, and then this is hot. I'm really sorry, guys. I'm not sure what the problem is. I already restarted the computer before the lesson, and it seems like it is still problematic for some reason. Please cope with me here. So we have the cold and we have the hot. And we will talk about the percent of saturated fatty acid. Okay, so remember when we drew the cell membrane, there are some phospholipids with saturated fatty acids, and then there are phospholipids with one unsaturated fatty acids, right? You can have both as unsaturated. It's kind of rare. So the question is asking you, if a plant lives in a cold environment, how much saturated fatty acids do you expect to find on the phospholipids? Do you expect to have more of it? Do you expect to have less of it? And why? That's basically what we are talking about. And here we have percent of unsaturated fatty acid. And finally, we have percent of cholesterol, okay, in the cell membrane. Okay, that's what we're talking about. So, cold. If the environment is cold, then things tends to solidify, right? Things tend to freeze up. Things tend to become solid. So we do not want the cell membrane to become solid. If the cell membrane is solid, then nothing goes through, right? So we want to keep it fluid, which means in a cold environment, you want to increase fluidity. Okay, so maybe we'll add uh, an extra column here. We'll say adaptation. Okay, adaptation is like how does it cope with the environment? And in the case of a colder environment, the adaptation we need is to increase fluidity. Okay, we don't want it to freeze up, so we need it to be more liquid. Okay, it's like the antifreeze in your windshield fluid. Right? We don't want it to freeze up when it's -20 outside. When you spray that stuff onto your windshield, you want it to stay liquid despite the cold, right? And that's what we're trying to do here. In the cell membrane, we want it to be fluid despite the cold environment. So how do you make it more fluid? Right? Check the notes from last time. Do you want more cholesterol or less cholesterol if you need the membrane to be more fluid? We wrote it down last time. I wrote it down in orange. I don't know what color you wrote it down in, but it's there. Take a look, please. Anybody? So it's over here. You can see more cholesterol means the membrane will be more fluid. And instead of memorizing it, let's try to understand why. When you have the cell membrane, it's mostly made up of phospholipids, and the phospholipids are packing close to each other, right? The more they pack close to each other, the more solid it's going to be. Right? We talked about this when we discussed saturated and unsaturated fatty acids, right? So if you stick some cholesterol in between, the cholesterols kind of prevent the phospholipids from coming together, right? They kind of get in the way. And if the phospholipids cannot get close to each other, then the membrane is going to be more fluid. So if the adaptation is more increased fluidity, you actually need to have more cholesterol, higher percentage of cholesterol. How about unsaturated fatty acid? If you need it to be more fluid, do you want more unsaturated fatty acid or less unsaturated fatty acid? Which one is fluid? Just think back the lipid stuff. Which one is liquid at room temperature? Unsaturated or saturated? Does anybody remember?
Unsaturated fatty acid is liquid.
That's right. So if you want your membrane to be more fluid, you want a higher percentage of this stuff. Okay? And you don't want too many saturated fatty acid. Okay? In a hot environment, the adaptation we want is lower fluidity, less fluid. Because if it's hot, everything is gonna be like moving faster. They're gonna be melting a little bit. So we don't want it to melt so much. If the cell membrane is like very liquid, then everything leaks out of the cell and things are gonna come into the cell. And we don't want that kind of unregulated movement. So the adaptation is lower fluidity, and to achieve that, you would have less cholesterol, less unsaturated fat, and more of this stuff, of the saturated. Okay, this is important. You should understand this for quiz number 2. Anybody have any questions before we move on? Okay. Next, we're going to talk about membrane protein functions. And again, I just want to remind you how when we drew the fluid mosaic model last time, there are proteins that are inserted in the cell membrane. Some might go through the entire cell membrane, some might be attached just on one of the layers. But regardless, there are some proteins that are in the cell membrane. And today we want to talk about the function of some of these proteins. So there are 6 functions that we will look at. Let's take a look at this overview diagram first. It's basically what we drew. The proteins that goes all the way through, okay, that spans from the outside of the cell to the inside. The inside, of course, is the cytoplasm. Okay, if it goes all the way through, then it's called an integral protein. Integral protein. If it's just attaching on one side, then it is called a peripheral protein. Peripheral protein. Okay. So, first function about these proteins. Number 1, attachment to cytoskeleton. Attachment to cytoskeleton. It allows cells to attach to extracellular matrix. So cytoskeleton we learned last time are these like actin fibers, microtubules, all these things that are kind of within the cell, but they're actually there are some cytoskeleton that exists outside of the cell as well. And in order to bind to these things, there needs to be some kind of anchorage point for them. Okay, and the proteins in the membrane, they serve as the anchorage points. Okay, so let's say this is the cell membrane. Okay, cell membrane, nucleus, and then you have all these cytoskeleton. Okay, when it gets to the edge to the protein, they need to be able to stick to something, right? To attach to something. So there's a membrane protein here. Oops, there's a membrane protein here. There's a membrane protein here. They basically allows these things to attach and provide support for them. Okay, and sometimes these attachment sites allows the cell to maintain a specific shape. Okay, such as those of the red cell. Now, this picture, all these lines that you see, they are all cytoskeleton that are outside of the cell. We have zoom out here in this picture. So that's a cell, that's a cell. All these things you're looking at, those are the cells. And all these fibers are on the outside. And in order for the cells to attach onto these extracellular matrix, they would need to use the membrane proteins as well. Okay. Number 2, cell signaling. Some membrane proteins, they are receptors, right? And a function of a receptor is basically to relay a signal from outside of the cell to the inside of the cell. Each type of receptor, they would have a binding site for a specific molecule. Right? For example, this receptor here, it would have a specific shape that would only allow these cone-shaped signals, molecules to bind to. If you have a molecule that's like a square, it won't fit. It will need a different receptor. Right? So different receptors require different trigger for them to be activated. Okay, so once the signaling molecule, should probably label that for you. This is a signaling molecule. Once a signaling molecule binds to the receptor, it will trigger a series of changes inside the cell. And depending on what kind of signal molecule you have, the kind of changes that are going on in the cell will be different. So in this example, we have adrenaline here. Okay. Adrenaline is it's a hormone that you release when you are excited, when you are in danger, right? The fight or flight response, that is driven by adrenaline. Adrenaline does have another name, like most people have heard of adrenaline, but then another name for that is epinephrine. And people who have allergies, right? What do they have to carry with them? Like kids who go to school, kids with severe allergies, they have to have anybody know what I'm talking
An
about?
EpiPen.
That's right.
EpiPen.
And yeah, the EpiPen, right? The EpiPen, the epi is epinephrine. Okay. So these are the same thing, right? So think of a person who's getting ready to run a sprint or some kind of race. They are going to need a lot of energy to power their muscle, right? So their body's going to release the adrenaline, right? And the adrenaline will bind to the receptor on a cell. And the receptor will cause a bunch of reactions to happen, which ultimately, in this particular case, lead to the hydrolysis of glycogen, right? You might remember glycogen is the storage form of energy in animals. We store them in muscle and liver. And when you do hydrolysis of glycogen, you release the glucose. So in this case, the adrenaline causes the release of glucose. Right? A different molecule binding to a different receptor is going to result in different outcomes. Right? But for all of these things to happen, all these changes to occur in a cell, you need to have some kind of membrane protein to relay that signal. Okay? So, and that's the second function of membrane proteins.
I have a question.
Sure.
So the molecule that's being attached, it doesn't actually go into the cell, does it?
That's a
Or
good question. It does not. Yeah, it just binds to it, and then it causes all the changes to happen. It doesn't physically go in. I actually this reminds me of like my childhood. My family does not like a Halloween, okay? So every year there's a Halloween. My mom would say, pretend no one's home. Okay, so we turn off the lights, but people still ring the doorbell. Okay, so when they ring the doorbell, my mom would always say, "Oh, quickly, hide in the basement, pretend no one's home." So we will all scurry down to the basement and then not make a sound. And then when she thinks people left already, then we can come back up, right? So this story reminds me of this situation because the kids who ring our doorbell, they never came into the house. But it causes all these changes inside the house, all these commotions going on, right? The house being the cell, right? All these changes is as a result of the signal being relayed through the receptor. right? The doorbell is the receptor. So maybe that story is going to help you remember some of these things. Now, we have lots of examples of these kind of signalings that are happening in the body. Another example is how neurons communicate with each other. And you will actually learn this next semester about the nervous system. Have you guys heard of dopamine? Anybody? Dopamine, what is that? No? No one heard of dopamine before?
Yeah, isn't it kind of like makes you happy? I don't really know how to explain it.
No, you explain it perfectly. This is the happy hormone, okay? When you do something that makes you feel good, your brain releases dopamine. You eat a cookie and it tastes good, feels good. You want another one later, right? It's the dopamine making you feel that way, okay? So dopamine will be released, and in order for the dopamine to be exerting its effect, it will have to bind to a receptor. Okay? Dopamine does not physically enter the neurons, but it will cause all kind of changes within the neuron that ultimately create that satisfying feeling that we have when we do things that we like. Okay, so that's an example, another example of cell signaling. All right, so attachment to cytoskeleton, cell signaling, and the third function of membrane protein is enzymatic activity. Okay, we know what an enzyme is. We learned that in lecture 2. They are proteins that catalyze chemical reactions. Okay, catalyze means to speed up reaction, right? Speed up reaction by lowering the activation energy, okay? By lowering the amount of energy required to cause a reaction to happen so that it can happen in the body. But a lot of times, we don't just do a single reaction and end up getting what we need in the body. In order to produce something that we need in the body, a lot of times you have to go through multiple reactions, one after the other. Okay? So membrane protein, in some cases, can act as enzymes. And more specifically, it would be multiple enzymes that are in a related pathway. So let's say we have these circle molecules and we need to change it to this hexagon molecule. Enzyme number 1 will convert it into triangle, and then immediately we can use enzyme number 2 to do another conversion and another conversion and another conversion. By putting these enzymes close to each other on the cell membrane, these reactions can happen rapidly in succession, one after the other. Okay? Instead of having the enzymes in the cytoplasm all over the place, right? Like you do one reaction here, and then now you got to wait for the molecule to be, transported to enzyme 2, and then to be transported to enzyme 3. Like it's just not very efficient, right? It's the same reason why we keep all our cooking stuff in the kitchen. Because when we cook, you need the chopping board, and you need the, pot, and then the grater. All that stuff is within the same vicinity because they are all needed for related tasks, right? You're not gonna keep the knife in the bathroom, right? Every time you need it, you gotta go run upstairs and grab it, right? So it's the same reason here. All these enzymes are for related reactions. You keep them all in the same place at the membrane, so you don't have to run around and increase wait time. Does that make sense? Any questions so far with 1, 2, or 3? Great. Number 4 is intercellular junction. We sort of talked about this when we discussed cytoskeleton. I told you at the time that there are places in the body where you want to create tight seal between the cells, so you're not going to have things leaking between them. The cytoskeleton can help with that, but the proteins, the membrane proteins, can also add to these kind of junctions, right? They are basically like hooks that you have, and anytime two cells having the same kind of membrane proteins, they would stick together and form a nice seal. This is the intestine, and this example, right, it makes sense to have these tight junctions because you don't want the food to be leaking between your cells. Okay? We will also have similar things like that in the blood vessels. Okay, you don't want the blood to be oozing all over the place either. Okay. Next, we have another function of membrane protein is cell-cell recognition. Okay? We drew the glycoproteins and the glycolipids last time. Let me just refresh your memory on what they look like. Hopefully, you still have the picture. Right? These glycoproteins are sugar stuck to proteins. Okay? It could be stuck to the integral protein. It could be stuck to peripheral proteins. But of course, the peripheral protein in that case would have to be on the surface. And we have sugar stuck on lipids, which creates these glycolipids. So collectively, these glycoproteins and glycolipids, they are important for forming an ID tag. Okay? And the ID tag allows your body to distinguish what belongs to the body, self, and what does not, non-self. When a bacteria invades your body, they would have very different ID tags because they have very different glycoproteins and glycolipids on their surface. So your white blood cells will be able to recognize them and attack them immediately. Okay, your glycoproteins, your glycolipids are going to be different from everyone else. Okay, you have a unique set of them. I mean, some of them will be similar or the same, but no two person would have completely identical set of these IDs. And that's why, like, unless you have an identical twin, okay? And that's why, like, you can't just take someone's kidney and then transplant it to any other random person, and they're going to reject the organ, right? And that is part of cell-cell recognition. Okay, it's the basis of how your immune system works. And again, you will be learning a lot about this next semester, okay, in lecture 3, actually. Finally, the last function of membrane protein is transport. Some molecules, as you will learn, will be able to cross the cell membrane on their own, no problem. But other times they might be too big or they might be charged instead of like, non-polar. Then they might have trouble crossing the cell membrane on their own. And that's when they need to rely on a transport protein to go into the cell or to leave the cell. Okay, so those are the 7 functions. Let me just quickly 6 functions. Let me just quickly recap for you. Attachment to cytoskeleton, cell signaling, and then we have the intercellular junction, okay, where 2 cells can stick together. We have enzymatic activity, cell-cell recognition, and transport. Okay, 6 different functions. I should clarify if the protein is doing transport, then it's not doing cell-cell recognition, right? And so like one protein is not going to be able to do all of these things, right? You need to have different kinds of membrane protein to do each one of these functions. Okay, let's see how you do on this poll question. It looks very complicated. So let me launch the poll. Even if you can't see the questions, I mean, if you can't see the poll, you can still participate. You can send me the answer if you want, in the chat box. So let's spend a minute on this. Okay, that was a minute and a bit. Anyone else before I end the poll? All right, we are looking for the true statement. So this is talking about the cell fluid mosaic model of the cell membrane, and specifically about the phospholipids. So phospholipids are free to depart from membrane and dissolve in the surrounding solution. That's not true. If that's the case, then the whole membrane would just slowly disintegrate and be completely broken down. They occur in an uninterrupted bilayer with membrane proteins restricted to the surface of the membrane. We know that's not true, right? Some membrane proteins can go all the way through. Some can be found exclusively on the inside. Doesn't have to be the surface. They have hydrophilic tails in the interior of the membrane. No, it should be hydrophobic tails. They can move laterally along the plane of the membrane. That's true. Okay, and then frequent flip-flop from one side of the membrane to the other. It's not correct. It's not correct. So the answer is D. That's the true statement. Which of the following is not a function of membrane proteins? Let's try this one. Let's keep it under half a minute. Go ahead. All right. Thank you to those who've answered the question. The correct answer is D. Okay. We talked about 6 functions, and D is not part of them. Right. The cell wall protects the cell from bursting, not membrane proteins. Okay. Next, we're going to talk about cellular energy. Cellular energy. Metabolism refers to all the reactions that are happening inside a cell. And we can separate the reactions into 2 categories. Catabolism, they are reactions where you break down molecules into smaller units. And in the process of doing so, it releases energy. Okay? So kata, the prefix here in catabolism, kata is related to the word catastrophe. In catastrophe, everything breaks down. So catabolic reaction or catabolism involves breaking things down. On the other hand, if you are building a molecule, if you're constructing a macromolecule using its subunits, then that is called anabolism. Okay? And that is going to require energy. Let's fill in this together. I think you have this picture in your study guide. And if you don't have your study guide, you can just try to follow along and maybe do a quick sketch of it, perhaps. So on the left side here, let me just zoom in a little bit for us. These are macromolecules. Macromolecules from food that we eat. And what kind of macromolecules do we eat? Give me some examples.
Protein?
Yep. So example proteins. What else? Maybe 2 more.
Carbohydrate.
Very good. Carbohydrates or carbs.
Lipid.
And lipids, right? That's good. So we don't deliberately eat like nucleic acid. They're just part of like living things that we eat, okay? But these are macromolecules. So after you eat them, after you consume them, you will have to break it down. You break them down into the many building blocks that they're made up of. Okay? And these include things like amino acid, maybe there are some glucose, okay, there are some glycerol, fatty acids, and whatnot, right? So all these are building blocks that can be used for biosynthesis. Biosynthesis is just like a fancy way of saying building things that we need in the body. Right? So because we are breaking these things down, the reactions are called catabolism. Catabolic reactions or catabolism. And in the process of breaking these things down, we are going to be releasing energy. Now, are we able to capture all the energy that are from the food? Actually, no, we are not too good at it. If you think about like a light bulb, like the old light bulbs, many people don't use these anymore. These are like the incandescent light bulb, the one with the filament inside. After you turn it on for, even 5 minutes, they feel very, very hot, right? So these are only about 10% efficient. Okay. 90% of the electricity that goes into the light bulb ends up being lost as heat. Okay, so these are very inefficient, only 10% efficient. And then we have the better light bulbs that looks like this. You might still have some of these at home. These are fluorescent light bulb, and they are much better. They are like 85% efficient. Okay, and nowadays, what do people use? Okay, it's fine. You might be using some of these as well. I still have
LED.
this in the garage. Sorry, what is it?
LED.
LED. And do you know how efficient LEDs are?
I think around 90.
Yeah, they're about 90% efficient, right? So even better than this, right? So now comes the question, what do you think about the human body? How efficient are we compared to a light bulb? Are we like the incandescent light bulb or are we like the LEDs? How efficient are we at converting the food energy into usable form of energy? What's the percentage? Anybody want to give it a guess?
25%.
5%.
25%, so precise. Did you see my note at the PowerPoint slide, Michael? But you're absolutely right. This is we are only about 25% efficient. A human body, it's about 25% efficient. Okay, and that means a lot of the energy from the food that we eat are lost as heat, which is fine because we need to keep our body at a core temperature of 37 degrees Celsius, right? So, when we eat, we feel warmer, right? We actually want to have those heat loss, right? Those are the heat that will help us maintain homeostasis. What about the energy that we can actually capture? And what are we going to use them for? Well, one way we can use them for is to take the building blocks and build things that we need. And when we build things, we have anabolic reactions. Okay, also known as anabolism. Okay, anabolism. Okay. And we can build various molecules that are needed in the body. Molecules needed in the body. And there are many things that we need, like hormones. Okay, muscle. Right? For example, the fingernails, they are proteins, right? But we don't eat fingernails to produce fingernails. We eat meat to get the protein. The proteins are broken down into amino acids, and then your cells at the fingertips they use the amino acids to build the fingernails, right? Yeah. So is this process of breaking down food that we consume so that we have the raw material and the energy to create what we need. Okay? Questions? Okay, so I'm just going to put a quick note here. The energy released, they are captured to produce a molecule called ATP. You can think of ATP as cell battery. Okay? That's what a cell uses to store energy. Right? Just like you charge up your batteries or whatever, right? You charge the battery of your phone. You charge those AA, AAA batteries so that you can use it in different electronics. Okay? The cell, we use something called ATP. Let's take a closer look at this ATP business. But before that, sorry, the main catabolic reaction that we use to generate ATP is once again cellular respiration. We roll it down so many times now, you should be familiar with it. And a part of it is going to be happening in the cytoplasm, and part of it is going to be happening in the mitochondria, and you will be able to generate quite a bit of ATP from the process, and we can use the ATP to power various things, including anabolic reactions. So ATP stands for adenosine, which is the same adenine as like the DNA adenine, adenosine triphosphate. That's what ATP stands for. Tri means 3. So in the molecule ATP, we have 3 phosphate group. The bond that's joining the last 2 phosphate is very energetic. That's where the energy is stored. So this ATP is like a fully charged battery. Okay, you can use it as is. And in order to use it, you need to break off one of the phosphate here. So the phosphate comes off and energy will be released. Okay? But once you break off a P, then ATP becomes ADP. So this is depleted battery. It's missing a P, okay? Adenosine diphosphate. But that's okay. You can recharge it. Right? When we eat food, when we do catabolism, the energy from the food, most of it is lost as heat, but some of it we can capture it. We can use it to recharge the ADP so that it becomes ATP again. Right? And this just goes back and forth all the time. Right? Anytime you need to use the ATP, you break up the P and then use the energy. Then you recharge it. As long as we are living, as long as we're alive, we can keep on doing this. So what do we use the ATP for? We can use it for various things like mechanical work, movement. Basically, mechanical work means movement. Okay. So this is a single cellular organism that has 2 flagella, and to beat them, that needs ATP. Same thing for humans, right? The sperm has a flagellum. To beat the flagellum, to move the tail, to swim around, that needs ATP. Transport work, to pump things across the cell membrane, sometimes you need ATP. Chemical work, to do chemical reactions, even with the help of an enzyme, sometimes you need to supply energy in the form of ATP. But regardless of what the functions are, regardless of what you're using the ATP for, what's common is that every time you use it, the ATP gets broken down into ADP, diphosphate, and a free phosphate. Okay. And with the food, when we metabolize the food, we can add this broken off phosphate back on and recharge them. Okay. Catabolic reaction, select all correct answers. We can try this one. Go ahead. Okay, that was a minute. They consume more energy than they produce. That's not true, right? The whole point of doing catabolic reaction is to produce more energy, right? So that we can capture them. They break down polymers into monomers, yes. They produce excess heat to warm the body, yes. And humans are not the only ones that do it, okay? All living things have to have some kind of catabolic reactions because one of the characteristics of living things is that they require energy. Okay, so to produce energy, you need to do catabolic reaction. All right. Next, we're going to talk about the types of transport. You know what? I think I'm just gonna push through and finish the lesson. I only have about 20 to 25 more minutes of stuff for today. Uh, we can just finish it and end early. So, types of transport. These are all red cells you're looking at. These are normal looking red cells on the left. These two are not looking too good. And so we're gonna talk about the conditions in which the red cells are going to be deformed like these. But first, let's talk about different types of transport. The first kind of transport is diffusion. You might have learned about diffusion in other classes before. It's basically the movement of molecules from an area of high concentration to an area of low concentration. For example, if you drop some dye into a flask of water, right, the dye is very concentrated here, but as time goes on, they would naturally spread out on their own. You don't even have to touch it, okay? So this is high concentration, this is low concentration, and naturally things will move from high to low concentration. Okay? Eventually, the whole thing will be blue. Okay? So this happens spontaneously. And the difference in concentration, it's called a concentration gradient. Okay, concentration gradient means a difference in concentration between two areas. And naturally, things will go from high to low concentration. If I spray some Febreze, like the air freshener Febreze, at the front of the classroom, eventually people at the back will be able to smell it. Okay? Diffusion just happens on its own. Now, there are different factors that affect how fast diffusion occurs. For example, things that are smaller tends to diffuse faster than larger ones. It's easier for small things to move around, right? Quickly, right? Have you ever tried to like catch a fly or something? It's really frustrating. Like they fly around so fast, and they are much smaller than us. They're going to be able to move faster. Similar concept here. If I have a molecule that's only 2 atoms big, like an oxygen, it's going to be able to diffuse faster than this glucose here. Okay. Second factor that affects the rate of diffusion, how fast is going to happen, would be temperature. Higher temperature causes faster diffusion because with higher temperature, the molecules have more energy. They are able to move quicker. That's why sometimes you will smell like, especially on like garbage collection days, right? Like you might be able to smell garbage smell very strongly on the street because it's hot in the summer, right? So all the smell is like diffusing out of the green bins. But on the winter, you barely notice. Okay, there's not a lot of diffusion going on. It's happening, but it's not happening at a high rate. Next, we have the steepness of concentration gradient. Okay, so concentration gradient, once again, it's like the difference between the concentration in two areas. So if the difference is a big difference, like here, I'm making these number up, okay? Like this is 100%, this is 0%. That's a big difference. And so these molecules are going to move over quickly. However, if the difference is not so great, let's say this is 80% and this is 40% maybe, totally making the numbers up. there is a difference. They will still move over, but it's not going to be happening, super fast. Okay. The last factor that we will talk about anyways, that affects the rate of diffusion is the charge between the two regions, okay, and the charge of the molecules that we're trying to move. So the thing here to know is that like charges repel each other. If you have things that are positively charged close to each other, the positive and the positive is going to push each other away. Okay, like these guys don't want to be next to each other, right? So it's going to push this thing over away. I'm trying to move it. Okay, it's going to move. It's going to move them away as quickly as possible, like that. Okay, so if they are the same charge and they're all aggregated in the close proximity, then the initial rate of diffusion is going to be faster. Okay, so 4 factors that affects the rate of diffusion. We have size, temperature, steepness of the concentration gradient, and the charge. Okay, please remember those. Next, we are going to talk about another kind of transport, which is called osmosis. Osmosis is the diffusion of water, basically. Okay, the movement of water across a selectively permeable membrane. Now, this is a confusing concept for some people, so let me try to explain this to you. Okay, don't look at the PowerPoint slide. Try to draw this out, and then I'm going to try to explain it to you. Okay, let's say you have a tube here. Okay, a U-shaped tube. Doesn't have to be U-shaped, okay, but I'm just going to draw it as a U shape so it looks like the notes and it will make sense to you when you look at it. And we are going to have a membrane here that separates the two sides. Okay, this membrane will only allow water to pass through. Okay, only allows water to pass through. Nothing else but water. So that's what we're going to do. We are going to fill up the two sides with some water. Okay, so I'm going to fill it up with water here. And I'll do the same, and I'll fill it up to the same level. There we go. So that's the water here. Now, on the left side, I'm going to dissolve some solute. And then I'll also dissolve some solute here on the right, but not as much. So these red balls, these are solute. Remember, solute is the thing that's being dissolved. And the solvent here is the water. Now, I deliberately draw the solute very big so that you can remember they cannot cross the membrane. Only water can cross the membrane. So the question you have to think about is which way will water move in this case? Will the water move to the right or will the water move to the left? Let me just do a poll and see what you guys think. My question is, which way will the water move? Remember, the red dots can't move, okay? If there's unrestricted movement, then the red dots can go to the right, right? They go from high concentration to low concentration, but they can't because I put a membrane here. So the only thing that can move is water. So which way will water move? If you think it's going to move to the left, please choose A. If you think it's going to move to the right, please choose B. If you think it's not going to move, please choose C. And please choose something if you're able to, okay? So that I can see what you guys are thinking. You can see a lot of your peers are actually not coming to class anymore. I remember we started with like 20, 23 people coming to the class, and now only 13 of you left. So, good job for coming to class. I have a lot of people saying it's A. Okay, so let's talk about this. Well, the concentration on the left here is actually high, right? High concentration. We can use square bracket to represent concentration. That's the official way of doing it. And over here we have low concentration. And since the red dots can't move, then the only thing that can help balance out both sides would be for water to shift. And what the water will try to do is they try to make both sides have the same concentration. And the only way to do it is basically for the water to move to the left. Right? You can think about why this works. Because as water moves to the left, you will have more water diluting the left side. Okay, so as H2O moves left, the left side gets diluted. While the right side gets more concentrated. Right? If you remove water from something, it gets more concentrated, right? That's how you like reduce like a sauce or something if you're cooking. Right? So eventually the water level is going to go up here on the left side and the water level is going to drop on the right side until the two sides are balanced. Okay. One way to illustrate this is to visualize what happens when you put a scoop of salt in your mouth and hold it. Okay. We can think about what happens. The salt is going to draw all the saliva out of your mouth, right? And that's a similar process as this. Water will move towards the side that has more concentration. Okay. That is osmosis. And that's how people, preserve food before invention of like refrigeration. Right? People salt their food. The salt basically dehydrates the food because of osmosis. And when there's no moisture, then the bacteria don't really grow, right? So salting is a great way of preserving food. But the salt is actually not too healthy for the body, right? Okay, so we're going to take one note and like a little bit more notes, and then that will be it for today. Okay, this is a little bit longer note. It's probably going to take a whole page, so maybe start a new piece of paper. All right, so here we are going to talk about the concept of movement of water across the cell membrane. Okay, and it's called tonicity. Tonicity. Okay. The definition is the ability of a solution to modify the volume of cells by altering their water content. So depending on what kind of solution you put a cell in, the cell is going to behave differently. Sometimes it's going to get bigger, sometimes it's going to get smaller, sometimes there will be no change. And that's what we're going to write down for this note. Okay. So let's start with the first situation. We have an isotonic solution. Okay, isotonic solution. Okay, this is before, and then this is after. Before and after, and we're gonna look at two situations. Okay, we're gonna look at what happens if you. This is a beaker. It's just a beaker, and I'm gonna put an animal cell right there. animal cell. And we will also look at what happens if we put a plant cell there. And remember, plant cells are more like rectangular in shape. We're not going to draw the nucleus, just going to draw the cell. Okay, this is a beaker, okay? Like a little glass cup. This is the solution, and this is the cell. Animal cell, plant cell. All right? So what is an isotonic solution? A solution is isotonic if the concentration of the solute, if the concentration are the same inside and outside, basically. Okay, now we're not counting the number of dots here, okay, because the outside has more volume, so you would expect to have more dots. But if you calculate the concentration, it will be the same. Okay? If it's 60% in the cell, it's going to be 60% in the solution. Same number, no gradient, basically. Okay, so an isotonic solution is where the concentration, okay, remember a square bracket means concentration, concentration of the solution equals to the concentration of the cytoplasm. Same concentration, no difference. So what do you expect to happen? Well, even with the same concentration, there will be movement of water. However, the amount of water that's coming in to the cell is going to equal to the amount of water that's leaving the cell. And that's the same for both situations, for both animals and plant cell. Okay, so the rate of water coming in equals to the rate of water leaving. No change in volume. No change in cell volume. One way to think about this is to imagine you have a basin and you have what do you call these things? Faucet. And then we're gonna turn on the tap. Gonna turn on the tap. Okay, so the water is gonna be coming in. It's gonna fill up the basin, the bucket, whatever you want to call it. All right, so water's coming in. And we're gonna poke a hole here. Gonna poke a hole here at the bottom, so water is leaking as well. So if you turn on the tap just by the right amount, the water coming in goes to the water leaving. What's gonna happen to the water level? Is it gonna change? Don't change, right? So that's basically what happens. Even though you're losing water, but water is also coming in, so as a result, there's no change. Okay, no change in the cell volume. So what happens to the cell afterwards? Okay, nothing. Nothing's gonna happen to the cell. Okay, so I'm just gonna draw the cell like this. Okay. I'm not gonna draw the dots anymore. It's just the same as before. And if it's an animal cell, it will be happy. Okay, if it's a plant cell, it's I mean, it's okay. It's not happy. It's not sad, but it's okay. Okay, okay means like it'll live, but it's not the best scenario for them. Okay. Questions so far? So I'm guessing your cells right now in your body will be in an isotonic environment. Okay? They're happy. Unless you are super dehydrated, you haven't been drinking water all morning, then maybe they're not isotonic anymore. Okay? So let's take a look at what the second situation is. Second situation is hypertonic solution. hypertonic solution. All right, so again, we have the before, we have the after, and we're going to draw the animal cell as well as the plant cell. There we go. You can label animal and plant if you want. In a hypertonic solution, there will be more solute outside, higher concentration outside than inside. Right? So concentration of the solution is greater than the concentration of the cytoplasm. Okay, the symbol is greater than, right? Well, you learned that in math, right? Here, I'll write down. So what happens? Well, which way is water going to be moving towards overall? Are you going to have more water moving out or more water coming in
More water going in?
into the cell?
Yeah, into the cell.
Okay, but remember, these black dots are not water, right? These black dots are not water. So let's go back to this picture here. The black dots are like the red dots. The dots don't move, but the water will move. Okay, so water is actually going to go towards the side that has higher concentration. Just like when I said putting a spoon of salt in your mouth, right? That's going to suck out all the water. Water will move towards the more concentrated end. So we would have more water leaving the cell than coming in. Does that make sense? Or you have a follow-up question, or anyone else of why this is happening? Okay. All right. So I'm just gonna keep it the same here. Before it would rate in is equal to rate out, and here rate in is actually less than rate out. The black dots are solute. Okay. I should probably label that for you. Okay. These black dots, they are solute. Okay. So like, sodium maybe, chlorine, so on and so forth. All right. So rate in is less than rate out. Let's do the bucket analogy to help you understand. So again, we're gonna fill up the bucket here. Okay, so the water is coming in, but this time we have a really big hole, so it's just like leaking all over the place. Water coming in and water going. So what's gonna happen to the water level over time? Any changes?
Down.
That's right, it's gonna go down. Water level is gonna go down. Right? So in the case of the cell volume decrease. Decreases. Okay? So our animal cell will shrivel up, shrink like that. You basically removing the water from it. It's like a grape. If you remove the water from the grape, it becomes a raisin. Okay? We have a name for this. This is called plasmolysis, which basically means the cell shrinks. Cell volume decreases, it shrinks. So for animal cell, it's okay. It's not ideal anymore, but they're gonna live. That's what happens when you don't drink enough water. When you're dehydrated, sweating a lot on a hot summer day for hours and not drinking, then your cell's gonna be shrivel up like that in the body. Um, and they're not gonna be happy. But you'll live. Uh, Obviously you need to rehydrate, right? Okay, but you'll live for the time being. And for plant cells, okay, the cell wall is still going to be like that, but then the actual cell will pull away from the cell wall, okay, and shrivel up on the inside. Okay, so there's the cell. This is the cell wall. Okay, normally the cell membrane is like directly touching the cell wall, but now it's like all shrivel up. And this is the cell. Right. Not happy. Right. The leaves are all wrinkled up and all shriveled. Not doing well. So this is isotonic. When your red cells are happy, looking good. It's like a jelly-filled donut with a little pinch in the center. This is hypertonic. Okay, look at how shriveled up they are. Okay, not good. Your blood is actually thicker than it should be, and you need to drink some water. You'll be fine though. Like you're not gonna, you know, be dying anytime soon. You just need to rehydrate. Questions before I show you the last scenario? Everybody happy? Okay. Last case, number 3. It is hypotonic. And I want you to write the O really big. Hypotonic. Okay, HYPO. Tonic. So let's draw it out. This is again before, after. So we have our cell. And in a hypotonic solution, you will have more solute on the inside than outside. Same thing for our plant cell. More solute on the inside than outside. Concentration of the solution is less than the concentration of the cytoplasm. Square bracket means concentration, okay? In case you missed it earlier. So what do you think? Do you think more water is going to be coming in or more water is going to be going out?
Out.
More water is going to be going out? Why is that?
Wait. So is there more stuff inside of the cell or outside of the cell right now?
Uh, is the picture confusing? I mean,
Oh, okay. So then the water would be going inside.
that's right, because there are more stuff inside, right? The dots are the stuff. Hey guys, for anyone else who's confused, the dots are the solute, the dots are the salt, if you would, okay? So you will have more water coming in to balance out the salt. Some water will still leave, but not a lot. So rate, I gotta do the same order. Before it was rate in is going to be greater than rate out. Okay, so in our bucket analogy, it's like you turn on the tap even more. Okay, so like turn on the tap to the max. And then the water is leaking. So because more water is coming in than leaving, the water level will go up. And eventually it will overflow, right? So in the case of our cells, rate in is greater than rate out, and the cell volume increases. And eventually the cell will burst. For animal, anyways. How come the plant cells don't burst?
Is it because of the cell wall?
Excellent, because of the cell wall. Okay, so this one is going to get too big, and then it's going to burst. Okay, lysis. That's the fancy word for breaking, for bursting. Okay. Needless to say, it's not happy. It's dead. But then for the plant cells here, okay, it just looks really, really puff. Okay, and it's going to be filled with water. Remember, the plants have like the central vacuole, right? We learned that last time. And the central vacuole will be filled with water, and it's going to love it. And it's going to be pushing on the cell. It makes it really, really, what's the word I'm looking for? Like a turgid, okay? It's very firm, and it allows the plant to stand up and open up its leaves, right? They love it. Okay. So remember, hypo, when you see the big O, it means the cell's gonna get really, really, really big. If it's animal cells, it's gonna burst and die. If it's plant cell, it's going to be all right. Is that okay? 3 scenarios. Okay, I think I'm going to stop there. I'll post this note. We will finish the rest of lecture 4. It's not going to take too long, probably like, 45 minutes, 50 minutes at most. And then I'm going to talk to you about the lab and how to do it, how to prepare for it. All of that will be on Thursday. All right. Thank you for coming. Enjoy the rest of your day. Bye for now.
Bye.
Bye.
Thank you.
No
Bye,
problem.
thank you.
Bye-bye.